Good morning, class, and welcome to Microbial Taxonomy and Diversity. So this is just only a continuation of your previous course in General Taxonomy, wherein under your Microbial Taxonomy and Diversity, we are going to focus more on the microscopic world of microorganisms. We are going to classify and identify all of those organisms from your bacteria, fungi, and even viruses. Okay. Now, to begin with, we will be using the different source or references. Any book we authored by Prescott et al. And then we have also Microbiology Principle and Exploration by Black. The third reference book is Microbiology, An Introduction by Tortora et al. And Textbook of Microbiology by Surender Kuma. All of these four textbooks can be downloaded in the internet. There are PDF copies of this book, which is free for downloading. You can use those ebooks as your references in the different activities and lessons that we are going to tackle under your microbial taxonomy. Now, let us first go back to the introductory part of taxonomy, which is the science of classification. Now, taxonomy is derived from an ancient Greek word, particularly the word taxis, which means arrangement, and the word nomia, meaning method. Okay? So the Greek word taxis means we are going to arrange organism, either arrange them by means of similarities or differences. While nomia, or the Greek word which means method, is how do we classify them? Okay, what method are we going to use? Okay? So there are various methods that are utilized in classifying organism, which will be discussed later on in our succeeding lessons. Now, taxonomy is a branch of science dealing with naming, grouping of organisms on the basis of the degree of similarity, and arranging them in order on the basis of their evolutionary relationship. Now, this particular method is a method that uses the old or traditional technique in classifying microorganisms. Traditional in the sense that we have a more modern method by means of identifying the relationship through DNA sequencing. Okay? That is the more advanced way using now what we call as genetic similarities in identifying the relationship of organism. But before we proceed to those modern methods, let us first go back and discuss the old method, or begin with the foundation on how taxonomy originated or started. Okay. Continuing, we have words that are related to our study, such as nomenclature, classification, and phylogeny. All of these words are not new to you because we have already encountered these words in your general taxonomy class. Okay. Now, taxonomy is very similar with other field of biology, particularly under botany, zoology, and also related sciences like physics, chemistry. Okay. So taxonomy is considered as synthetic in the sense that it is man-made, and it is a multidisciplinary science. When we say multidisciplinary science, it involves other branches of science in the study in classifying organisms. Okay, so there is physics involved in taxonomy, there is chemistry involved in taxonomy, and there is biology involved in taxonomy. Okay, moving on. Now, in taxonomy, we owe its progress on the advancement made in other branches of science like morphology, histology, physiology, cell biology, biochemistry, genetics, molecular biology, and computational biology. So when we say advancement, the integration of these other field of sciences made taxonomy a more broader as well as more enjoyable field of science. It is a combination, like what we have said a while ago. Okay, so when we say morphology in taxonomy, uh, it involves the discussion as well as identification of morphological similarities. So morphology means more on the structure. Okay, so when we talk about morphology, we talk about how a particular microorganism is shaped or how it is made up. Okay, so morphology is more on describing the appearance, particularly the shape. Okay, so if you can still recall, in your microbiology, we have discussed different morphological shapes of bacteria, such as cocci, bacilli, spirili, and others. Okay, so those are morphological characteristics. And then also taxonomy involves histology. So when we say histology, it is the study of cells. Okay? So microorganisms are also made up of cells. Although your viruses, they are acellular in nature. As compared to your fungi and bacteria, viruses are different with regards to their cellular structure because they are acellular in nature. So when we say acellular, when they are inside a host body, they are alive or active. But when they are outside of a host, they are not considered as a living organism. Okay? That's why they are classified as cellular. We do not say that they are dead when they are outside the host, but instead they are not active when they are outside of a host cell. Okay, but when they enter a particular host, they become active. So, for example, if an individual sneezes or cough, the droplet coming from the nose or the mouth is either transferred via air or it will become airborne and then it will settle down on any hard surface. Now, the virus that is present on those droplets, they become inactive because they are already outside of a cell. They are inactive, but they can still become active once that they again enter a living host. Okay, that's why there is a need for us to sterilize our equipment or any object that is contaminated with any kind of droplet. That's why we are advised to continuously use use alcohol in order to avoid acquiring those different types of viruses, especially nowadays during this time of pandemic. Okay, so moving on, we have another branch of science that is involved in the study of taxonomy. We have physiology. So a while ago, we discussed about morphology, which is the shape or structure. The next one is physiology, which is the function. So morphological structures have their own specific functions, and that involves physiology. We will study the physiological function of each of the organs within the microorganism. So within a microorganism, there are various cellular structure or morphological structure present, like your mitochondria, your ribosomes, your Golgi apparatus, and others. Those different cellular structures within a microorganism have their own specific physiological function. That's why physiology, which is another field of science, is associated directly in the study of taxonomy. Okay, and then we have cell biology because different microorganisms are made up of different types of cells. Okay, so we are talking about the cellular structure of these microorganisms. And then also it involves biochemistry or the chemical components of a microscopic organism. Okay, so we are made up, or microorganisms are made up of carbon, hydrogen, oxygen, nitrogen, or the basic chemical components of life. And biochemistry come into the scene when we are going to study those chemical components of microorganisms. And then also we have genetics. We are going to discuss how these microorganisms multiply, how they transmit their hereditary information or DNA, and at the same time, how does mutation occur? Okay, so it will involve the genetic study, which is closely associated with another field of science, which is molecular biology. Okay, so your molecular biology involves now the study on the genetic material that is within a microscopic organism. For example, in molecular biology, we are going to identify how a particular virus mutates and what this particular mutation will eventually become into. Okay, so when a virus mutates, it will again restructure its genetic or the DNA into another structure wherein it will not be identified by your uh, body or by the antibodies. Okay, so antibodies that are being developed by the body will eventually not be able to detect viruses that have undergone mutation because these viruses have changed in their molecular structure. And then the last field of science associated with taxonomy is your computational biology. Under your computational biology is we are going to discuss computation with regards to normal levels of certain biochemical components. So when we say normal levels, what is the normal level of the glucose? What is the normal level of your lipids, etc. That falls under your computational biology. Okay. Now, for classification purposes, organisms are usually organized into subspecies, species, genera, families, and higher order. If you can still recall the hierarchy of orders. But anyway, we are going to discuss again those particular organizations of life for as a review. And for eukaryotes, the definition of species is the ability of similar organisms to reproduce sexually. So eukaryotic microorganisms reproduce sexually, while most of the time your prokaryotes do not undergo sexual reproduction. Eukaryotic prokaryotic microorganisms usually undergo asexual. But with regards to your eukaryotic microorganism, most of them under or reproduce sexually with the formation of your zygote in order to produce an offspring. Okay. Then, aside from taxonomy, it is closely associated or related to systematics. Okay. So, when we talk about systematics, it is the study of the diversification of living forms. Remember, we came from an ancient ancestor, meaning man and ape. We are not directly connected or not directly our ancestors are not apes, but instead ape and man originated or evolved from a common ancestor. Okay, so when we say evolved, the our ancient ancestor, which is similar, or our ancestor, we came from a one ancestor. Okay, so galing tayo sa isang ancestor. Okay, so under your systematic, we are going to go back in how microorganism diverse. Okay, how did they evolved? Are they similar with regards to their ancestry? And if they are similar with regards to ancestry, how close are they to one another? Okay? So this is where systematic will come in. So we are going to understand both past and present relationship among living things, particularly microorganism, because you are going to focus more on bacterial or microbial taxonomy. Okay? So again, we are going to go back and study the evolutionary trees, wherein under your evolutionary trees, we have again your synonyms, cladograms, phylogenetic trees, and phylogenies. So I know you're going to say that you have already taken this one in your general taxonomy, yes. But as a form of review and uh, as a form of enhancement from your previous lesson, we are going to discuss again those particular terminologies under your systematics, particularly those areas under your cladograms, phylogenetic trees, and phylogenies. Okay. Then your phylogenies have two components. We have the branching order, which usually shows the group or relationship of microorganism from one another. And then the other component is your branch length, which shows the amount of evolution. Okay? Which particular microorganism is younger, which particular microorganism is older. That is the meaning of your branch length. Okay. Next, we are going to discuss once again your phylogenetic trees of species and higher taxa, which is very commonly used in the study of evolutionary traits, such as your anatomical or molecular characteristics, as well as the distribution of organism, or what we call as your biogeography. So when we talk about biogeography, in other words, you are going to identify the location of the microorganism. Is it present in colder regions or warmer regions, or is it present in a certain environment only? Okay, so is it global, gaya ng COVID, which is already considered as a pandemic type of viral infection? Pandemic in the sense that it infects the whole world or the whole population of the planet. That's why it is pandemic. But if it only infects a small area or a particular area, it is only considered as an epidemic. But with regards to your COVID-19 virus, it is already pandemic because it is all throughout the globe. Okay? And then your systematics, in other words, is used to understand the evolutionary history of life on Earth. Okay. Now, we are going to discuss also the different aim or goals of taxonomy. Okay? Our main goal or objective or our main focus will be on the classification of microorganism, followed by identifying and studying the relationship among those organisms. And the third objective is to provide the universal identification of a microorganism. Okay. So next, another topic that we're going to discuss for today's slide is on the classification. Okay. So when we say classification, it is the orderly arrangement of organism into groups. So when we say orderly arrangement, it means that we are going to classify them from the highest taxa and then until the smallest unit or lowest unit of the taxa, which is the species. Okay. So in other classification method, they usually include the domain. But in our microbial taxonomy, we will start with the kingdom. Okay? We will not anymore include the domain, but we will start our orderly arrangement beginning with the kingdom. Okay. Now, there is nothing inherently scientific about classification, and different groups of scientists may classify the same organisms differently. Okay. So sometimes it is better for us to utilize scientific names rather than using common names or local names. Because if you can recall, in different places, they have different common names or local names they use to identify organisms. Okay? Now, when we are going to classify, particularly microorganism, we will not utilize the common name or the local name. Instead, we will utilize the scientific name for a more subjective as well as more formal way in identifying a particular organism. Okay. Now, sometimes different groups of people or scientists may classify organism in a more different way. Okay, for example, your clinical microbiologists are interested in the serotypes. These are the numbers associated with certain microorganism. Okay, so each microorganism, as they mutate or evolve, they produce a certain serotype and this serotype will become or converted into numbers to easily identify them. For example, our flu virus have thousands of serotype. So it means even if you have a flu vaccine, that particular vaccine will not be effective, especially if that particular type of vaccine is only specific for a certain serotype group of flu virus. Okay, so let us imagine that your flu vaccine is serotype 0101, but the flu virus that infected you is serotype 0202. So it means that the vaccine that was injected to you will not be effective because the virus that have invaded your system is not the virus directly associated with the vaccine that was injected to your body. Okay, so ganon yung how clinical microbiologists identify microorganism, yeah, even bacteria have their own various serotypes. Okay, these particular serotypes can also produce resistance patterns, and also they are associated with the production of toxins or poisons. Once an organism or microorganism enters our body, it will produce certain toxin that will cause now the activation of our defense mechanisms, such as the production of mucus, because the body will try to protect the mucosal lining of your lungs as well as your nasal area by producing excessive volume or amount of mucus in order to wash or to block the invading microorganism. And sometimes this toxin may also trigger what we call as fever. Once you have a fever, it means that the body is trying to kill the microorganism by increasing your body temperature. Usually, microorganisms such as bacterial infection usually live or grow in temperatures that range from 30 to 37 degrees Celsius. So 30 to 37 degrees Celsius is the most adaptable temperature for bacteria and even viruses to multiply or grow. So the body will increase its temperature from the normal 36 to 37 to a higher temperature which will exceed the normal body temperature, such as 39 to 40 degrees, in order to kill the invading microorganism. But sometimes uh, the excessive body temperature may also lead to other negative effect to the body. Okay, it may cause convulsion, and also very high temperature may affect the brain itself. It will not only kill the bacteria, but it will also affect the neurons in the brain. Okay, so fever that is still within the range of 37 to 38 is still considered as okay because the body is fighting the infection. But when your body temp starts to rise from 39 going higher, there is already a need for intervention, such as taking in antipyretic medicines, such as those medicines that will relieve or decrease the fever. Okay, so those are some of the examples on how different field of science may classify or identify organisms. Okay, going to the next slide. Now, under your classification, we have three types of systematic, or how we are going to study taxonomy. One is we are going to study first the evolutionary systematics or grouping of organism, particularly by identifying their common ancestors. The next type will be your numerical taxonomy, which will involve the use of mathematical models to group organism according to overall similarities. And then the last type of systematics or taxonomy is the phylogenetic classification system. So wherein we are going to group based on their similarity and evolutionary relatedness. Okay, so those are the three types of systematics or taxonomy. Okay, and then for identifying, it is a practical method or use of classification criteria that we are going to use in distinguishing certain organism from each other. So when we are going to identify an organism or microorganism, we are going to verify the authenticity first. Verifying the authenticity involves the searching in the internet. Then we are going to identify also the strain of the microorganism, the reaction of the organism to certain chemical compounds, and what is the proper or best way to isolate and identify organisms that causes a disease. The next slide tackles with the history on nomenclature and taxonomic hierarchy. So if you recall, in our previous lesson in general taxonomy, the father of modern taxonomy is Carolus Linnaeus. Okay? He was a Swedish botanist known as the father of taxonomy. And it was credited to him the establishment of taxonomy as a separate science. Now, he was instrumental in framing the rules for naming the organism, which he applied uniformly while giving his classification. Now, if you recall, there are other people or scientists who classified organism, but the method that was introduced by Linnaeus is a more systematic method. That's why we are still using the taxonomic hierarchy that was devised by Linnaeus. Now, it was he who popularized the binomial nomenclature, that is the modern scientific way of naming organism. So when we say binomial nomenclature, we will start from the domain, the kingdom, and then going down to the lowest level, which is the species. Okay. Next, the binomial nomenclature, like what I have said a while ago, is composed of two parts. Okay, so a binomial nomenclature of a particular organism will always have its genus and the species. Always remember that the genus is always in a or it is written always in a capital letter, and then the specific name will always be written in a small letter. Okay. Now, using the scientific name is universal, meaning it is used in different regions or countries. Okay. Also, when we classify organism, we always either use Latin or Greek. But most of the organism are classified using the Latin language. Although it is already considered as a dead language, it is still used in science, particularly in naming or classifying organisms. Now, nomenclature of organism is governed by set of rules framed by international codes of nomenclature. In other words, you cannot just name a particular organism. For example, you accidentally discovered a new plant variety or a new species of animal or any type of microorganism. It doesn't mean that you can name that discovery automatically. It will undergo what we call as uh, or it will follow set of rules. Okay, so commonly in each domain, for example, or group of microorganisms such as bacteria, animal, and plants, there is a specific governing body in naming or classifying those discovered organism. For the discovery of new strains of bacteria, we have the ICNB, or the International Code for Nomenclature for Bacteria. And then for zoological nomenclature, we have the International Code of Zoological Nomenclature, or the ICZN. And then we have also the International Code of Botanical Nomenclature, or ICBN. Okay, so zoological pertains to animals, while your botanical pertains to plants. And then we have also your International Code of Zoological Nomenclature, or ICZN. Okay, so nadoble lang. And then we have the last one, the International Code of Nomenclature for Algae, Fungi, and Plants, or the ICN respectively. All of this International Code of Nomenclature are responsible in properly naming and classifying discovered organisms. Okay? Now, always remember that when you are going to write a particular scientific name or when it is cited in text, it should be written either in italics or underlined font style. Okay? Now, usually the name of the author who first gave the correct name as per rule is written in abbreviated form, and the specific name is written in Roman. Okay, that is how we are going to write scientific names. Next slide. Okay, so that ends our topic for this morning. The next slide contain your enhancement activities. Okay, so there are guide questions. So there are 15 guide questions that needs to be answered. So you are going to place your answer and upload it in our Google Classroom. So you're going to place your answer in either in Word format, and then you're going to upload it in our Google Classroom for checking. Okay. So submission of your activity or enhancement activity will be until Friday. Okay, I will give you time to go over the different questions and to answer them. Okay, so submission will be until Friday, October 15. So again, thank you for listening, class. See you on Friday. This coming Friday, we are going to discuss your microbial taxonomy activity number 1, or the laboratory portion of our course. Okay, so there are, as you can see on the slide, there are 15 questions. So read each questions carefully, analyze the questions before you answer and submit on our Google Classroom. If you are going to submit on the Google Drive, send me the link so that I can access your answer. Okay. So have a rainy day or stormy day. Take care always. See you on Friday.